We have learned about stem cells coming from different types of tissues within your body, and that they're involved with repair and regeneration and normal maintenance of tissues and systems within your body. However, to fully appreciate the role of stem cells, we have to also understand their implications in cancer. In biology, cells die by normal mechanisms, and it can be via two different ways. Apoptosis is programmed cell death. This is executed in such a way by the body so as to dispose safely of cell corpses and fragments without eliciting an immune response. However, if a cell dies within the body by necrosis, it causes an uncontrolled release of antigens and causes inflammation. This is usually due to injury. However, there is a balance between cell division and apoptosis, and there is a role in disease. Too much apoptosis, we have tissue atrophy, neurodegeneration, and too little, we now have cancer, where cells grow out of control. The idea of cancer cells arising from a common origin has been thoroughly explained and published. It states that cancer is part of the healing process, and that the disease only manifests itself if its control are impeded. The old cancer cell model shows that all tumor cells can form new tumors, and therefore equally tumorigenic. Unregulated growth is due to serial acquisition of genetic events leading to the expression of genes that promote cell proliferation with contaminant silencing of growth inhibitory genes and blunting of cell death. Cancer is a proliferative disease. However, the new cancer model shows that tumors arise from cells termed cancer stem cells that have properties of normal stem cells, particularly self-renewal and multipotentiality, but unregulated cell growth is due to a disruption in the regulatory mechanism in stem cell renewal. Cancer is a stem cell disorder and not simply a mechanism whereby a cell proliferation is disrupted. These cancer stem cells persist in tumors as distinct population that likely causes relapses and metastasis. This theory explains why cancers are so difficult to treat. The issue is, are we targeting the right cells? Conventional chemotherapies kill differentiated or differentiating cells, which form the bulk of the tumor, but are unable to generate a new one. A population of so-called cancer stem cells, which gave rise to it, remains untouched and may cause a relapse of the disease. Development of specific therapies targeted at cancer stem cells holds hope for improvement of survival and quality of life of cancer patients, especially for sufferers of metastatic diseases where little progress has been made in recent years. With the old cancer theory, we see that chemotherapies are able to kill the differentiated cell types but leave the stem cell untouched, and therefore the stem cell, or cancer stem cell, can regrow the tumor again. If we're able to design drugs that are able to kill the cancer stem cell, then the tumor will regress and not relapse. Tumor heterogeneity provides evidence for cancer stem cells. Most tumors are very heterogeneous, and heterogeneity is commonly retained by tumor metastasis. This implies that the cell that produced the tumor had the capacity to generate multiple different cell types, which is a classical hallmark of stem cells. The next step is to figure out what makes the cancer stem cell different from other cells in the tumor. Stem cells are more difficult to kill because they are so important throughout a person's lifetime. They have developed mechanisms to protect themselves. Drugs that are developed to target cancer stem cells may represent the most significant application of stem cells in medicine. To date, determining the differences between normal stem cells and cancer stem cells has produced few hits. Fundamentally, the difference between normal and cancer stem cells still remains unknown. Ironically, the cancer stem cell theory represents the largest barrier to commercialization of stem cell therapies. The cancer stem cell theory has placed huge demands for preclinical testing, tumorigenicity testing on stem cell-based therapies. and it is this close association of normal and cancer stem cells that has significantly increased the risk of such treatments. Although we have described that cancer stem cells can be the origin of tumors, we also have to understand that stem cells can be used to treat cancer as well. This is an example where we use a prodrug approach in combination with a stem cell in order to treat brain tumors. One of the classical activities of stem cells is that they have the ability to migrate, or a tropism, towards tumor tissues. In this case, stem cells engineered to express a prodrug activating enzyme is used. Stem cells will then track down the tumor within the brain, and in combination with the prodrug are able to kill the tumor. Here in the schematic diagram, we show that neuroblastoma tumors are injected into the mouse and induced in the mouse. We then have a neural stem cell line that's been modified to express the enzyme that converts the prodrug to the active drug. These stem cells track the tumor cells down and bind to them. The prodrug is then added, and at the site of tumors, the drug is converted into the active form, where it then kills the tumors. These studies were pioneered by Karen Aboudi, where she was able to generate a human neural stem cell line expressing the prodrug conversion enzyme and bring it forward to clinical trials in the treatment of inoperable brain tumors or recurrent high-grade gliomas.